Welcome to the deep dive. So today we're jumping into a really fundamental topic.
The fundamental topic, really.
Right, we're looking at the cell cycle, and we've got a whole stack of sources here. Everything from basic cellular biology all the way up to some pretty complex systems engineering.
And that's really the mission for this deep dive. We're trying to move past just the textbook diagrams.
Yes, the simple circles.
Exactly. We want to understand the elegant logic behind it all. We'll be connecting these abstract models, things like coupled oscillators, directly to cancer therapy, and even how scientists physically measure this process.
To see not just what happens, but why it's so incredibly reliable.
It's all about structural guarantees.
Okay, so let's start with the absolute basics. We're talking about the eukaryotic cell cycle, which has 4 main phases, and the first 3 are all kind of bundled together into what's called interphase.
Right, and interphase is anything but a resting state. It's incredibly active. It starts with G1.
The first growth phase.
Yep. The cell is bulking up, duplicating organelles, cytoplasm, basically everything except its DNA.
Because the DNA comes next, in the S phase, for synthesis.
It's a critical step. This is where the cell replicates its entire genome. So for a human cell, you start with 46 chromosomes, and you end S phase with 46 duplicated chromosomes.
Each one now looking like a little X with 2 sister chromatids.
Exactly. And then you have G2, which is the final quality control check.
Making sure that all that new DNA is perfect before the big split.
Perfect, and making sure all the machinery for the split is ready to go.
And that split is the M phase, mitosis and cytokinesis.
Mitosis is the nucleus dividing, separating those duplicated chromosomes. And cytokinesis is the cell itself pinching in two,
Creating 2 identical daughter cells.
And it's worth just a quick mention that the system is ancient. Prokaryotes, they use a simpler version called binary fission.
But the principle's the same.
The principle is the same, and the protein they use to do the pinching, FTSZ, is actually homologous to our tubulin, the stuff that makes up our mitotic spindle.
So this idea of precise division has been around since the beginning.
Forever.
And that precision is really the key, isn't it? I mean, replicating billions of DNA base pairs perfectly.
Yeah.
It sounds like an incredibly high-stakes operation. How does the cell guarantee it doesn't make mistakes?
It's not just a nice to have, it's mandatory. And it does it with these intense quality assurance stops. We call them checkpoints.
Decision points.
Exactly. Where everything just halts until specific conditions are met.
So let's talk about the first one, the G1 checkpoint. It's often called the restriction point. Why is that one so critical?
Because that's the point of no return. At G1, the cell is doing a full assessment. Is it big enough? Does it have the energy, the raw materials?
And most importantly, is the starting DNA damaged?
That's the big one. Because once it passes this checkpoint, it is committed. It has to finish the cycle. If it fails the check, it can either pause to repair
Or it gets out of the cycle completely.
it enters a state called G0.
Okay, so if G1 is the commitment check, what's the purpose of the G2 checkpoint later on?
G2 is the final pre-flight checklist before mitosis. It is a hyper-detailed scan to make sure every single chromosome was copied perfectly during S phase. No damage, no missed spots.
You cannot enter M phase with a flawed instruction manual.
You cannot.
And then there's a final gate, the M checkpoint or spindle checkpoint. And this one happens right in the middle of mitosis itself.
Right at the end of metaphase.
Why there? What's it trying to prevent?
It's preventing a catastrophe. The next step, anaphase, is when the sister chromatids get pulled apart.
And that's irreversible.
Completely irreversible. So if even one of those duplicated chromosomes isn't attached properly to the spindle fibers from opposite poles,
The daughter cells get the wrong number of chromosomes.
which is a condition called aneuploidy, and it's almost always a disaster for the cell, the M checkpoint literally stalls division until that alignment is perfect.
So this whole system sounds like it needs, well, accelerators and brakes. Let's start with the positive regulators, the accelerators.
The main players here are 2 types of proteins. You have cyclins, and you have cyclin-dependent kinases, CDKs.
And the name CDK kind of gives it away. It's a kinase, so it phosphorylates other proteins to activate them, but it's dependent on a cyclin.
Exactly. The CDKs are sort of always there, just waiting. They're the stable engine.
But the timing, how does that work?
The timing comes from the cyclins. Their concentration levels go up and down dramatically throughout the cycle.
So they act like a clock.
A clock and a sensor. For example, cyclin B levels shoot up right before M phase. When cyclin B binds to its partner, CDK1, they form an active complex. You might see it called MPF.
Maturation promoting factor.
Right. And that complex just triggers a cascade of phosphorylation, driving the cell right past the G2 checkpoint and into mitosis.
But of course, a good system needs a powerful failsafe.
Yeah.
So let's talk about the brakes, the negative regulators.
The tumor suppressors, absolutely essential. And the most famous is probably P53.
The guardians of the genome.
That's what they call it. If P53 detects significant DNA damage, it slams on the brakes. It can halt the cycle and call in repair enzymes.
Or if the damage is too severe,
It triggers apoptosis, programs cell death. It basically tells the cell to self-destruct so that damaged DNA can never be passed on.
and then there's the other big one, the retinoblastoma protein, RB. It's the gatekeeper for that G1S jump, right?
It is the gatekeeper. RB works by literally grabbing onto and inhibiting a key transcription factor called E2F.
And E2F is what turns on the genes for DNA replication.
Correct. So as long as RB is holding on to E2F, the cell is locked in G1. But as the cell gets signals to divide, CDKs start adding phosphate groups to RB.
Phosphorylating it.
Right, and this phosphorylation weakens RB's grip until it's hyperphosphorylated and just lets go of E2F.
And then E2F is free to rush off and turn on all the S-phase genes.
And the cell barrels past that restriction point.
That decision point in G1, it leads us right to the next section. Because you mentioned if a cell fails the check, it can exit the cycle and enter G0. Now, we often hear G0 called the resting phase.
But our sources make it really clear it's not that simple. It's anything but passive.
So what is it then?
G0 is a state outside the normal replicative cycle, a quiescent stage. But there are at least 3 different flavors of G0.
Okay, let's start with the most flexible one, reversible quiescence.
This is what you see in tissue stem cells. Think muscle stem cells, or even mature liver cells, hepatocytes. They're in G0 doing their job, but they can be activated to start dividing again if there's, say, tissue damage.
They're on standby.
Exactly. And one of the sources brought up this really neat idea of a GLERT phase.
Alert, so they're not just waiting passively.
No, the idea is that they actively get into a state of readiness. It's controlled by a sensor complex called mTORC1. By keeping mTORC1 at a certain level, the cell is primed. It's ready to go, skipping some prep steps if an injury happens. It's active preparedness.
That's a huge contrast to the second type then, irreversible G0.
Or terminal G0. This is the permanent exit. These are cells that are so specialized mature neurons, skeletal muscle, that dividing is just not an option. It would destroy their function. They're never coming back to the cycle.
And then the third type, which is clinically very important, senescence.
Senescence is also irreversible G0, but it's triggered by severe stress. Usually things like telomeres getting too short or a dangerous oncogene getting activated.
So it's a safety mechanism.
A critical one. It's an alternative to apoptosis. The cell doesn't die, but it is locked down permanently so it can't replicate and cause harm, like becoming cancerous.
So if one of those reversibly quiescent cells gets the signal, what's the molecular trigger to get it out of G0 and back into G1?
It's all about phosphorylating RB again, but the initial players are a bit different. It's mainly a complex of cyclin C and CDK3.
So cyclin CCDK3 is like the first alarm clock waking the cell up.
That's a good way to put it. It specifically phosphorylates RB at 2 sites, S807 and 811. That causes RB to release E2F.
And E2F then kicks off transcription for G1 entry genes.
And crucially, it also fires up the massive production of rRNA needed to build new ribosomes. You need protein factories before you can start growing again.
This level of precision, this on-off switching,
Yeah.
it really starts to feel less like biology and more like engineering.
That's where the sources go next. They shift to looking at the cell cycle through the lens of dynamic systems theory.
The idea of a coupled oscillator model.
Exactly. The cycle, especially in something like budding yeast, behaves just like a non-linear bistable switch.
A latching-gate. Okay, so what does bistable mean here?
It just means the system has 2 and only 2 stable states it likes to be in. Think of a light switch. It's stable when it's on, and it's stable when it's off.
So for the cell, one state is G1.
Right. G1 is a stable state maintained by high activity of a complex called APCC.CDH1, which cleans out all the mitotic proteins.
And the other state is SG2M.
The SG2M state, where CDH1 is turned off, allowing all that division machinery to build up.
So to get from one stable state to the other, you need a specific,
A transient push, a pulse of activity. To get out of G1, you need a pulse from a transcription factor called MBF. To get back to G1 after mitosis, you need a pulse from a protein called CDC14.
And once you get that push, the system latches into the new state.
It snaps into place.
And this is where the math comes in. The model uses two coupled van der Pol oscillator. That sounds intense.
It sounds complicated, but the concept is actually pretty intuitive. Think of the cycle as two competing modules. You have one oscillator, call it by 1, that represents the G1S module driven by MBF.
In the other.
The other, by 2, is the MG1 module driven by CDC14, and these two modules mutually inhibit each other. When one is on, it actively shuts the other one off.
So the math just formalizes that biological feedback loop.
Perfectly. And in the model, the difference between their activity, Y1 minus Y2, is a direct analog for CDH1 activity, the master switch.
Okay, so when you actually solve the equations for a normal healthy cell cycle and plot it, what does it look like?
This is the amazing part. When you plot the trajectory on a graph of MBF versus CDC14 activity, you get this beautiful, distinct butterfly shape.
A butterfly.
Yeah, with two wings or loops. One loop is the cell firing up the G1S module, then it switches, and the other loop is the cell firing up the MG1 module to reset. That butterfly shape is the mathematical signature of a healthy, alternating mitotic cycle.
The math proves the biology. So what did the math predict happens when you break the system?
This is where it gets really powerful. The model shows if you increase the inhibitory coupling between the two modules, that's the B variable in the equations, you break the latch.
You break a butterfly.
You break the butterfly. The system collapses from those stable double loop oscillations into a single pathological loop, an endocycle.
Which, in a cell, would be something like endoreplication, where it just keeps copying its DNA without ever dividing.
Exactly. And the model even predicts the exact mathematical tipping point. It's called a homoclinic bifurcation. It's the precise threshold where the elegant controlled system collapses into chaos.
That's a huge leap. To know that the stability of life is governed by dynamic systems theory. So, let's connect this back to the real world. A broken latch in a human cell.
That's the definition of cancer. It's fundamentally a disease of a broken cell cycle.
Yeah.
You either have accelerators stuck on oncogenes or the brakes have failed faulty tumor suppressors.
And because we know the specific molecules, they become targets for therapy. We hear a lot about CDK46 inhibitors.
Drugs like palbociclib, ribociclib, very common in treating certain breast cancers.
And their mechanism is aimed right at that G1 checkpoint.
Precisely. They work by blocking the hyperphosphorylation of RB. So RB never lets go of E2F. They essentially reinforce that G1 lock, stalling the cancer cells and preventing them from dividing.
So that's the application. But on the research side, how do scientists actually measure this? How do you look at a tumor sample and see where the cells are in their cycle?
The standard technique is called flow cytometry. It's actually very clever. You take your cells and you stain their DNA with a fluorescent dye.
A dye that binds stoichiometrically.
Meaning, the more DNA you have, the brighter you glow.
Ah, so you can tell by the brightness.
Exactly. You run the thousands of cells, one by one, past a laser. Cells in G0 or G1 have a normal amount of DNA, so they create a big peak of low fluorescence.
And cells in G2 or M have double the DNA.
So they create a second, smaller peak at exactly twice the fluorescence intensity. And the cells in S phase, which are actively replicating, show up as a smear of cells in between the two peaks.
It's the quality of that data must be critical, though.
Oh, absolutely. You need sharp peaks. Scientists measure the sharpness of that first G0, G1 peak with something called the coefficient of variation, or CV. You want a CV under 3%. If it gets too high, your peaks are too fuzzy, and you can't tell G1 cells apart from early S-phase cells.
It's amazing how this topic, which seems so fundamental, is actually conceptually really difficult.
Yeah.
The sources pointed out that even biology student teachers get it wrong.
It just highlights how hard it is to teach a dynamic process with static pictures. One of the biggest misconceptions, and it just won't die, is that interphase is the resting phase.
When it's actually the busiest time for the cell.
It's where all the work happens. Growth, replication, it's incredibly active.
Another common mistake is the timing of replication itself, isn't it?
Yes. So many people think DNA replication happens during prophase, which is part of mitosis. But it only happens in the S phase, strictly.
And then there's just the terminology. Centrosomes, centromeres.
Ventrioles, they all sound the same, but they are completely different things. It's easy to get them mixed up.
So we've really gone on a journey here, from the basic phases to the molecular breaks and accelerators, then all the way to the beautiful mathematics of the butterfly trajectory, and finally back to the clinic.
It really hammers home that latching-gate concept. The cell cycle isn't a smooth, continuous process. It's a series of stable states, and you need these specific transient pushes to move from one to the next. That's what makes it so robust.
And that's why catastrophic failure is the exception, not the rule.
So as we wrap up, here's a final provocative thought from the sources for you to think about. We focused a lot on CDK46 inhibitors working at the G1 checkpoint. But some research now suggests they also suppress tumor growth during G2 arrest by preventing something called stress-induced endoreplication. This suggests that the connections between these safety checkpoints, G1 and G2, are more complex than we thought, and it really makes you wonder, how many other unknown safety mechanisms does the cell have ready to slam on the brakes?